Diatoms are a major class of unicellular algae in the phytoplankton. These organisms have a silicified cell wall composed of two overlapping halves, called epitheca and hypotheca. It is estimated that diatoms produce between 20 and 30% of the oxygen we breathe. They are at the base of the trophic pyramid in the oceans and have traditionally been considered an optimal food for zooplankton larval growth. However, at the end of the 1990s was discovered that a few diatom species produce oxylipins, molecules that derive from the oxygenation of the fatty acids by lipoxygenase activity, and that can induce abortions or congenital malformation in the copepods, that are the main herbivores of zooplankton. The phenomenon is a chemical defense due to grazer-induced synthesis of oxylipins that are not detectable in intact diatoms. So, synthesis of oxylipins is committed to reduce the grazing impact of subsequent generation of copepods. This in order to allow diatoms blooms to persist to their pressure. Lipoxygenases, or LOX, are the enzyme responsible for the oxidation of polyunsaturated fatty acid to oxylipins. This reaction takes place in the LOX catalytic pocket in presence of an iron ion as cofactor and a water molecule. The iron cofactor is usually coordinated by 5 residues, 3 histidine, 1 asparagine, and 1 isoleucine. Iron ion extracts a hydrogen atom to the nearest carbon of the substrate, at 2 positions upstream or downstream of the oxidation of the double bond. This according to the substrate orientation in the pocket. So, if the substrate polar head is located externally to the active site, the oxidation takes place 2 positions after the carbon nearest to iron. Otherwise, if the tail is oriented to the external side, 2 positions before. This happens due to the fixed position of the oxygen channel in the structure. So, according to the position of the double bond oxidized, it is possible to have different LOX types also for the same substrate. The aim of the project have been the search for putative LOX sequences from diatoms and their functional and structural analysis in order to obtain a classification and a characterization of putative LOX from diatoms. To achieve these goals, bioinformatics tools have been used, involving transcriptomic and protein sequence database search, sequence and clustering analysis, model constructions, also improving AlphaFold models, and docking study by both flexible and rigid protocols. By searching all database, 45 possible LOX from diatoms have been found. Thus, starting from an optimized multiple alignment of all sequences selected, a clustering analysis has been performed by the construction of several phylogenetic trees. And here you can see our best result obtained. From this phylogenetic tree, it is possible to detect at least 6 different groups. For each one, we choose a representative protein sequence that we have modeled in the active conformation, so in presence of both water and cofactor. All groups, except for the first and the second, differ in the iron coordination residues, in particular in the fourth, that could be substituted by histidine, glutamine, or serine. Moreover, group 3 present a deletion between the first 2 histidine of coordination, while group 4 and 5 show an insertion between the third and the fourth coordination residue. For each representative sequence of the group, docking analyses have been performed with EPA, the main fatty acid present in diatoms, or with other suitable substrates selected from the literature. Results have shown that different residues in the active site pocket result in different LOX types, making different interaction with the substrates. For example, for what concerns the first 2 groups, where no difference in terms of coordination residues have been detected, it is possible to see as the behavior of the 2 enzymes is different. In particular, for the LOX from Skeletonema marinoi, we performed docking analysis not only with EPA, but also with other 2 substrates detectable from the literature. Results have highlighted as EPA interact with the iron by the carbon 7, and since the polar head is in the external part of the pocket, the enzyme behavior is typical as a LOX 9. Instead, the other 2 substrates bind the cofactor by the carbon 11, but their polar head is oriented in the internal part of the pocket. So, also in this case, the enzyme behavior is typical as LOX9. Looking at the binding energy, we can see that there is a preference for these LOX for EPA and esa-trienoic acid. For what concerns docking of LOX from pseudonichia renicensis with EPA, it is possible to see as LOX type is LOX12, due to the extraction of the hydrogen from EPA carbon 10. The pocket composition shows two aromatic peripheral residues, according to other LOX12 structures that are available in the protein data bank. Binding energy and constant of inhibition are comparable to redocking study on LOX12 crystallographic complex, for which results are not shown in this poster just for space reason. These results are also in agreement with literature data, which report a possible rule of LOX from pseudonichia renicensis as LOX12 or 15. So, main difference between group 1 and 2 is not attributable to a difference in the cofactor coordination, but to the presence of a specific motif along the sequences of the second group that is absent in the first group, and vice versa. In conclusion, we hypothesized the presence of 45 uncharacterized proteins with lipoxygenase activity in diatoms that can be divided in at least 6 groups and for which the LOX type was detectable. As a future perspective, we would like to extend our work to other substrates or to other novel LOX sequences in order to enhance the analysis of pockets composition in diatom LOX.